The land cover classes, the habitats which the research was done, it was done in the grassland, acacia, savanna, mosaic, lava forest sparse, which was sparse, the cloud forest, woodlands, and the woodland which was thick woodland, whereby a total of 415 assembly plots were established, whereby apart from the cloud forest and the lava, the other areas, the plot had a radius of 17.84, but within the lava and the cloud forest, the radius was 10 meters, the distance. Before the training was done, the people were supposed to conduct the survey. They were trained. Those were the field botanists and the samplers within the region. And during the survey, people used standard operating protocols. The team which participated in the study, they include staff from museum, Kenya Forest Research Institute, University of Nairobi, and University of Eldoret. During the data collection, the field data collection, each group comprised of 4 biodiversity samplers and 1 botanist, whereby they were following the standard operating procedures to undertake the biomass. And the data was captured in a special hub. You see the smart hub and the GPS. That's a picture of people who are undertaking the survey. And that is a picture of the botanists and the biodiversity samplers setting up a plot. After the study was done, the biomass data was not analyzed. What was done was we were after the plant biodiversity, which was analyzed using SPSS, diversity index, and the relative indices were also captured during the data analysis. And from the research, the area generated a list of 250 species. We were mainly dealing with the trees, not another herbaceous plants or climber. So trees and the shrubs. So a total of 250 species was generated, whereby the DBH of the plants which were being captured were shrubs or trees who had a DBH of 5 centimeters DBH at a circumference of 15.71. And from the species captured, there were, out of the 250 species captured, 170 species were under the IUCN Red List status. 4 species were endangered, 4 were vulnerable, 7 were near threatened, and 155 were of least concern. Those are the species which were in the IUCN and the ones which were in CITES. 7 species were listed in CITES. The ones which are shown there. For the floral occurrences, 10,900 trees within the shrubs were captured within the 415 assembly plots. That 4 assembly plots were not in existence. And of the project sites, the Chulu Hills National Park had the highest plant diversity, followed by Kibwezi, each with the number of species, 332 and 317 individual species. And out of that, it means that, given that the 2 parks are protected, that's why the diversity was high as compared to the other areas like Kuku and Rombo, which are community ranges whose diversity was less. That was Kuku A and the Rombo Ranch, which had less data. For the frequencies of the families, plant families, a total of 41 families were recorded within the 250 species, whereby Fabaceae, the legumes, were the most dominant, followed by the Baceracea, Sapidacea, and Celastracea. On the other hand, Anisotraceae, Canelacea, Guttevera, and Icacines and Philanthacea are the lowest species within the study area. On terms of the genera frequencies, the top 8 genera were the Commiphora, Acacias, Diospyros, Pepris, Combretum, Trichocladus, Olea, and Dobea, while Wabugia was the least dominant family within the Assembly region. For the species distribution, the topmost occurring species were Diospyros, Combretum, Schumannii, among the rest. On terms of species diversity, the overall Simpson Diversity Index was determined as 0.9. Species diversity indices per area are represented below, as you can see, with the Chulu Hills areas at the highest species diversity as compared to the other regions within the study area. However, as earlier mentioned, species richness was highest in Chulu Hills National Park and the Kibwezi Forest. From the research, there are some things which the research or the project was able to capture, whereby they were able to capture a lot of data. Robust data was generated from the research. Fossil specimens were collected. There was capacity building from the different institutions which were undertaking the work. There was build-up of taxonomic skills for botanists, and the basic botanical skills acquired by the biodiversity assemblies, and the RED Plus project partnership was strengthened with other stakeholders. The challenges the team faced during the work, there was an issue of accessing the assembly plots or the area they were undertaking the research. Since that it was not a vast research to be done, there was another one. The one which was done was just to satisfy another research which was done. There was an issue with getting to the plots which were earlier laid. The weather was not favorable. Identification of the dead plants, given that biomass is in all plants, it was an issue when it identified the name of the species, since it had no characteristics for a quick identification of the plots. There was an issue of wildfires and the sensitive plant species, because some of the areas we found they had experienced quite a lot of fire. So, conclusion and recommendation from the research. According to the research, we found that there was need for further survey to include the grasses, the sedges, and the other herbaceous plants, since the research only captured the trees and the shrubs. There was need to do a monitoring plan. It was required to ensure that the assembly plots are well managed, and then expansion of partnership in the future with the project, since the experts were not they are not partners of the project. And the data generated can be used in monitoring of effects of climate change and biodiversity within the ecosystem. And there was need for more inventory in other key biodiversity areas of conservation importance in the country. Thank you.
Thank you, Judy. We are still moving, and we are now going to the next presenter, and she will present on the red list assessment and biodiversity data as a foundation for the defined key biodiversity areas, insights from using flowers in Namibia. It will be by Lovisa Kadenge. So remember, we'll have questions will be at the end of the presentation, or the presentation as we agreed before lunchtime.
Good afternoon, everyone. My name is Lovisa Kandenge, and I'll be presenting on Red List data as a foundation for key biodiversity area identification, insight from the endemic flora of Namibia. I am coming from the National Botanical Research Institute in Windhoek, Namibia. So, background of our plant biodiversity, Namibia has about 4,000 indigenous plant species, of which about 700 species are endemic to the country. So in recent days, many of our species are facing increasing environmental and anthropogenic pressure. And without scientific evidence, we can't support conservation decisions. We can't use assumptions and opinions to come up with conservation decisions. And that's why there's a need for scientific assessment tools which can provide us with scientific evidence to support conservation decisions. There's a lack of baseline data which resulted in many of our important plant areas being left out of protected areas. Back then, we mostly only focused on animals, and we somehow neglected plants. Continuing with the background, so the identification of KBAs is being driven by two projects, which is being run under our institute. The first one is the SBAB project, which focuses on the entire country, and this is where we mostly do the IUCN Red List of our endemic species. So I'm only focusing on the endemics, but we also include the near endemics for the whole country, and it's the main driver for the KBAs. And then we have the second project, which And then we have the second project, the GRS project, which is in the southeastern Namibia, the bounded with the red outline. And this project came in talks because this area is under-collected and under-documented. So this area is an important area to Namibia because it's one of the areas with the special habitats. These are types of habitats that certain plants can only grow in, not anywhere else. And yeah, they are called Quartz Island. Okay, so why are we doing this study? We want to evaluate how Red List assessments can feed into the identification of key biodiversity areas in Namibia. And the assessments, we want to focus the assessments on endemic flora, which will become our trigger species for the key biodiversity areas. And we also want to identify spatial and taxonomic data gaps. So the endemic regions of Namibia, we have two main regions, one in the northwestern, of Namibia, that's the Kaukofeld Center of Endemism, and one in the southern region, which is the Succulent Karoo Center of Endemism. So With the JRS project, we are trying to see if we can also delineate the Varambat in red to become a center of endemism, because it holds about 36 currently identified endemics, plant endemics. So why do we need red list data? It provides us with the threat status and the extinction risk of a plant species. And it also supplies us with information on locality and population data, which later on helps us do mapping, species range mapping and species monitoring. And it's also one of the critical key, if I'm using the right term, for identifying KBA trigger species. So our red list progress. We have a total of 887 plant species which are assessed and published on the IUCN of threatened plant species, on the IUCN red list of threatened species, not plants, sorry. And out of the 887, 229 are endemic. And so we started trying to see which assessments are outdated, and we are trying to update them. So currently we have 70 endemic species reassessed and published, which this started in 2023. And out of those 70, 64 are threatened, and an additional of 32 assessments are on the way, in progress. They are not yet published, so they will add to the 70 which are reassessed and published. And in overall, we have, overall of all the species, we have 42 species which we have mapped their distribution. And so said, we still have 471 endemic species which remain unassessed. So moving on to the main thing, key biodiversity areas. I'm sure most of us knows that these are globally important sites for biodiversity, and they are identified using the IUCN Global Key Biodiversity Area criteria. And they are mostly based on threatened and restricted range species, which then helps support protected area planning and conservation prioritization. So right now, at the moment, we don't have any key biodiversity area in Namibia. We only have legacy sites, which were identified in 2001. And we have a total of 21. And these were triggered by birds and aquatic invertebrates. Plants were not included. I'm sure you are probably wondering why there is a legacy site down here, which is a center of endemism for plants. This is because this is also a heritage site. And it's kind of a habitat for certain types of migratory birds. So I believe that's the reason why. So also, you can, so we need to review this legacy site and see if we can keep them and identify them as our new key biodiversity areas, or we can downgrade them and, delist them. And as you can see, there is no legacy site identified in the Kalkofeld Center of Endemism. So that's an issue. So we are trying to identify new key biodiversity areas which will at least include most of our plant endemic areas. Methodology. So we have been doing this through field surveys, where we go to areas which are under-collected, or we know that these areas have endemics. So we go there to do monitoring, and then we do plant collections, and we take population data, and we also do photographic records for citizen science, iNaturalist. And then after field surveys, we move on to the herbarium. So this is where we do identification, specimen digitization, and then filing and putting records on our database, which is called BRAMS. And then with all the information from field surveys and herbarium records, then we carry on to do our red list assessments, whereby we apply IUCN red list categories and criteria, and which will later give us the status of each species that we assess. This will then help us map distribution for each species, and with all the combining all the four processes, yes, then we can come up with KBA scoping, which we apply the IUCN KBA criteria, and then we identify trigger species, and only then can we delineate potential KBAs. So our key findings, on the map, there are polygons with a green boundary. So those are our protected areas. So I've included them in the map so you can see how they are overlaying with our trigger species. So the map is basically showing all potential trigger species that we have identified from the assessments. So this potential trigger species includes range restricted, and it also includes the threatened species. Not every range restricted is threatened. Okay, so we have, I've mapped, it's a map that I've created myself. I've mapped 6,002 records of trigger species, and many of the records occur outside protected areas. And then I later on had to filter, and I wanted to map out the threatened trigger species only. And those summed to 2,890. And as you can see, there's a high concentration of threatened trigger species on the southwestern of Namibia. Although there are still other trigger species throughout the country, most of them are concentrated here. And you can see the ones below here. This is where our JRS project comes in. That's why we want them to, we are trying to make it a key biodiversity area also. So these are just examples of our potential trigger species. So all these species here, they have been assessed, and they are all threatened. Most of these species, you only find them in the dry area. Yeah, as the map I was showing you the distribution of the threatened species. So this you can only find within that range. So we have Allodendron, Dichotomum, Adenia pechueli, this is critically endangered. We are facing a big issue with poaching of Adenia. And we have Hoodia alstoni, Avonia quinaria, Lithops juli, and Schwantesia constanzi, and Crassula corallina. So major threats to Namibian flora, our biggest threat to Namibian endemics is climate change, because most of our endemics are found in the desert and in dry areas. We have been experiencing a prolonged drought since 2015 up to now. It's still ongoing. And then we have habitat fragmentation and transformation. This is coming from mining and development, and we have a green hydrogen project that is about to start in our let me just go back. In this, that is going to start in this area. It was implemented to be in this, or planned to be in this area where most of our threatened and endemic plants this is our center of endemism, with a high concentration of threatened plant, yet this is where that project is going to be. Okay, so and then we have illegal collection of succulents. I was just talking about poaching now, and we have overharvesting or overexploitation. We also have an issue of timber, overharvesting of timber species in the northeastern of Namibia. So in the picture, I'm basically showing that this lithops species used to occur here before the habitat was disturbed. And as they were developing the area, constructing a road in this area, they had to clear the area, so the population of this species was lost. And then in the pictures, I'm basically showing you plants that we took from poachers. We confiscated from poachers. So up there is Adenia. It's like almost every single month we are having a case of Adenia being confiscated by a border post, or reports of someone who have seen an individual poaching it. And then down here, I have included Welwitschia pictures in the presentation, although Welwitschia is not endemic to Namibia, but it's a range-restricted species. So we share it, so it goes until Angola. So these bags here, these are cuttings of Welwitschia. These are 50 kg bags that we confiscated from poachers. And then when we took them out of the bag, this is how they looked. So we also have an issue of Welwitschia seeds being poached. These are two different, this is connected to this, but this is another incident of poaching. So data gaps and challenges. We have so many under-collected remote regions. On the map, it shows the ones with the blue, purple, those ones are the areas which are very well collected. And as the color changes from blue to yellow to white, then the yellow and the white, those are the under-collected areas. So we are trying to fill the gaps. So, and we also have limited range maps for endemic species because we don't have enough data or enough field data to do range maps for most of our endemic species. We are running behind with our red list assessments. So we have incomplete red list coverage, and also we have issues with taxonomic uncertainty. With all the taxonomists coming and telling us, no, you should call this that, it's not acacia, it's Vachelia, or it's Senegalia, so our assessments are being affected by that. And then we also need, we have a need for updated georeference occurrence data, especially for data that was collected back in the 90s. Most of the scientists or botanists, they only used to collect, and the locality record that they'll put there is a center of a QDS, which does not give you the exact locality of a record. So in conclusion, Namibia's endemic flora is very important globally, and Red List is a foundation of key biodiversity area identification. So with the findings that can support evidence-based land use planning, and which will balance economic it will balance economic development with biodiversity conservation. So in the end, I can say if we don't have data, then we can protect our biodiversity. So thank you. That's at the end of my presentation, and I would like to thank all the sponsors and funders.
Thank you, Olofisa, for that, our next presenter is Beatrice, who will give us a presentation on digitization of and publishing of museum specimen to GBIF. Welcome. Are you using this?
Then you can disconnect.
Okay, fine.
Good afternoon. I hope you are not sleeping. Okay, I'm Beatrice Mokami, and I want to present you a project titled Digitization and Publishing of DTCD Specimens from the National Museums of Kenya Invertebrate Section or Invertebrate Collection to Global Biodiversity Information Facility, a part of a larger project funded by JRS, and in collaboration with Kenya Methodist, where I did my bachelor's, and also to appreciate the AKE, NMK again, and Sajorek for the sponsorship for the conference. Welcome. To start with, the TCD is a family of beetles in the order Coleoptera. They live in fresh water, and that's the picture of the DTCD in question. They are very critical in ecological, they play a very critical ecological role because they are formidable predators. They feed on other aquatic invertebrates, therefore bringing ecological balance. They are also bioindicators, whereby they can be used to indicate the quality of water or freshwater systems, and also they can be used as biocontrol for they feed on mosquito larvae, which are aquatic, therefore helping to control malaria and mosquito population. The gap or the need to do this project is because DTCD data has been unavailable in global digital databases, therefore hindering comprehensive ecological studies. For like the previous presenter said, with no data, there is no protection, and with no data, no research that can be done, or less research can be done rather. Also, this has limited the biodiversity monitoring, and as well, conservation planning has not been well done because of lack of this data. Sorry. The objectives of this project were 5. I don't know what has just happened. One was to extract the data from labels of these specimens from the collection, clean that data, put latitude and longitude, which are the coordinates, validate the data, and finally publish it on GBIF. Materials needed, just a laptop or a desktop. Catalog numbers for museum specimen, it was necessary to have the catalog numbers for unique identification. Scissors, specimens themselves, stable internet for the cleaning and validation, power, and myself, of course. The methods to start off was data extraction, whereby drawers were retrieved from the collection, and then specimens retrieved from cabinets, and then the information was keyed in on an Excel sheet. Before going to the next, this data is not enough for, it's not standard to be published on GBIF. So some other key information was, there was need to add that information, which was gotten from the internet and then fed on the Excel sheet. Later, that Excel sheet was loaded into an Open Refine tool. This helps to fill in the missing information, more missing information, and also standardize these fields. An Open Refine is better used for larger data sets. If you have fewer records, cleaning can be done manually. But in case of museum specimens, since they occur in large numbers, it's better to use Open Refine tool, which helps to clean the inconsistencies. It gives you a platform where you can see and correct errors on the platform, harmonize names, and many other corrections that are necessary. The other method was georeferencing. As Lovisa has said, collections made in 1990s and earlier years did not come with coordinates. Therefore, there is need to put coordinates on them. And on this section, I used the Geolocate Batch client. Is a tool that assigns coordinates to the textual locality information. It generates a map, like the picture, and then the records can be verified manually to ensure that the points are better placed. Once the records have the correct coordinates, the data is then put into a data validator, which is the GBIF data validator. This as well flags errors and also in other inconsistencies. For example, the scientific names. Unlike the Open Refine, it doesn't offer the platform to make corrections there, but after flagging, for example, those ones in yellow, then you can go back to your Excel sheet and make corrections. It also highlights the coordinates that are not well placed, and by that you can go back to Geolocate and ensure that they are well placed. After the data is valid, all is well. By the help of the NMK node managers, it was uploaded on the NMK integrated publishing toolkit. Metadata was created. I was helped by my supervisor, Laban Joroge, and then eventually the data was published in GBIF. This is the link to the data set. It was a part of a larger data set, so it's not the only one. It is part of another aquatic data. Results. At the end of the project, 1238 records were digitized and successfully published on GBIF. 16 genuses were identified. 101 records were identified up to family level, meaning they had no genus or species name. And then the temporal range was from 1912 to 1993, a range of 81 years. This is the distribution map of the DTCD. In Africa, 1237 out of the 1238 were African. Only one that came from Europe and in specific Hungary, but this is believed to be a sign of historical exchange, or historical specimen exchange, not that it's the only one that was recorded in Hungary. These are a map showing the temporal range. You can see there was a spike in collection around mid-1920s, 1925 to 1930. And probably it's because of the political instability that was happening then. You know, that's when the First World War was First and Second World War happened. This to show that it is important to keep sampling, because if you were to do comparison with DTCD data from 1960 all the way to 1980, you can tell that we have nothing from the museum to compare from. In conclusion, 1238 records were published on GBIF. The gap of African DTCD data was addressed. The Kenyan contribution to global biodiversity informatics was strengthened, and finally, freshwater ecosystem research enhanced. Recommendations. I would recommend expansion of digitization efforts, since, for example, from the invertebrate collection, we have over 3 million specimens, and less than 50% are the only one that has been digitized. And we said that having these records not digitized means less conservation efforts. So I would recommend to expand digitization efforts. Another one was to identify the old specimens. From the results, you have seen that 101 records were unidentified to species level, meaning the older specimens that are already preserved on the collection has not been, some of them has not been identified to species level. Therefore, it is necessary or good to identify them. And lastly, but not least, capacity building in biodiversity informatics is necessary, and its enhancement is key. That's all. Thank you.
Thank you, Beatrice. We have 2 more presentations, and the next one is by Angeline. Is Angeline in? Presenting on flower visitors and species into some coffee species in Taita use. Sorry? So she not today. Okay, fine. Thank you. So then our last presenter, and remember, if I had said that you keep your questions, because after the last one now, we'll have questions and answers from all the presenters. So let me welcome Edward. We will present on fish species of the Mara River and threats.
Good afternoon. My name is Endon Jagi, and I'll be presenting on the fishes of the Mara River. This is what you find on our website, so you can note this so that you can get more information about this project thereafter. And we did this work together with the University of Florida, the Randolph-Macomb College, the US, and funded by the JRS Biodiversity Conservation. Okay, the Mara River, as you all know, is one very, very important river in the country, or in the Lake Victoria Basin, mainly because it supports the life in the Mara Serengeti ecosystem. Actually, without the Mara River, we would have no Masai Mara, and we would have no Serengeti. The two reserves actually generate a lot of income in the East African countries, Kenya and Tanzania. So that tells you that the Mara River is a cross-border shared between Kenya and Tanzania. The source is the Mau Forest Complex in Kenya, and comes in from two sources through two tributaries. One of them is called the Nyangores tributary, and the other one is called the Amara tributary. The Amara tributary comes from a source called the Paraket in the Mau on the northern side, and the Nyangores tributary comes from a source called Enapuipui on the western side of the Mara forest. So the river is roughly 395 kilometers from the Mau forest to the river mouth in Musoma, Tanzania. Along the way, it is joined by several tributaries. One of them is called the Tarek River. The Tarek River comes in just before the Mara River leaves Kenya for Tanzania. But high above is another small tributary coming from a small catchment in Kenya called Ngare Ngito. So the Tarek River comes from Loita Hills in Kenya. But the base flow in the Mara River mainly is from the two major tributaries that I just spoke about. Now, this study was divided into three areas. One of them, what we're calling the Upper Mara, is actually in or near the Mau forest, and that is quite high above. The first site was on the Amara side of the tributary. The second site was on the Nyangores River, actually in the protected part of the Mau forest. The third and fourth sites actually were slightly below the Mau forest, in areas that have smallholder farms. So that is what we are regarding to as the Upper Mara. So the Middle Mara actually is where we have a multi-branch, and going down actually to the Mara, Masai Mara National Reserve. Both sites 6, 7 are in the Mara. Then we choose one site in the Tarek River because it's a very, very important tributary in that it brings a lot of input into the Mara River. So the lower region actually basically is in Serengeti and the lower parts of the Mara River, including Site 10, which is majorly affected by a lot of mining, utilizing a tributary called the Tigithe River. Those that have been to Tanzania, this is where the Nyamongo mines are located. So site 11 and 12 are majorly in the Mara wetland, because the Mara River flows into Lake Victoria through a massive wetland called the Mara wetland, that also serves as refugia and breeding areas for fishes of Lake Victoria around that part of the lake. So, if you go to literature, you read that there are around 40 species in the Mara River. Actually, those are also records that are available at the ichthyology collection here at the museum. But our study found around 38 species. That is not to say that the two other species are not in the river. probably because of timing of the survey period and all that, we never captured. But it also doesn't mean that there are only 40 species. There could be more. Actually, more work in this year could produce more species. And in fact, looking at this, because these are all species found in the Mara River, we earned several species of Enteromias that we could not identify to species level. We've sent them out for genetic analysis, and that probably could yield more species that could be new in science. So, looking at the abundance throughout the span of the river, Enteromia species, actually from number 1 to around 6, will tell you that they occupied almost the entire length of the river. So normally Enteromia species are what we call survivors. They can survive in any kind of environment, including very harsh environment. Now, in the span of 395 kilometers of the Mara River, these candidates were found all over. And looking at this, especially this, the Crarias, or the African catfish, or the mandyfish, was found all over. This candidate also survives very harsh environments, but definitely you'll find them more abundant in good water quality. This candidate here, called Ningu in the local name, or Lake Victoria labeo, and Labeo victorianus, was initially categorized as critically endangered. But through our studies, because apart from the Mara River, we also looked at the other rivers draining into Lake Victoria. And being an endemic species to the Lake Victoria region, it was actually classified from critically endangered. And if you now visit the IUCN Rain list, it's actually a fish of least concern. It is very, very abundant in the rivers. Other fishes actually that were found across the length of the river were these ones you can see here. This is Enteromias jacksoni, very distinctively known because of the 3 black spots. Actually, even new ichthyologists will recognize this. Cynodontis, these are what we call the squeakers. This is also endemic to the Lake Victoria region and was found across the length of the river. Others were Oreochromis niloticus. This is one of the introduced species in Lake Victoria region, now spread all over the country, across Kenya, and a very good candidate for aquaculture. So you find a lot of this in the farms throughout the country. Now, in the upper part only, and not in other part of the river, were these 2 species of the same genus, the Clarias. This particular one likes inhabiting actually marshy areas. It's called the Clarias aluandi, and you only find it in this part of the country, the Lake Victoria region. Clarias rheocephalus, as opposed to Clarias aluandi that likes swampy and marshy areas, this one likes running waters. So in most cases, you'll find it into the river system itself, actually even in the middle of the river. The middle part only, this is where we had Masai Mara. You can note actually the biomass of the specimens that we caught. This particular one was 8 kgs. Basically, no disturbance, no fishing, nothing happening. The Momiras, I'll be speaking about this much later because it's one of the candidates that is affected by massive fish die-offs in the Mara. You've read that in the papers enough times. Coptodon here, most people confuse this for the Nile tilapia or Oreochromis niloticus, but the difference is very simple. These broad bands in the middle and then black spot at the dorsal fin. So in the lower part only, the lower part mainly comprised of the Mara wetland and the parts just above the Mara wetland. And this is the lungfish. This one is very well known by probably most of you. We only have 3 species of this genus in Kenya, one found in the Lake Victoria region, the other one found in Lake Turkana, and the other one found in the Tana Delta. Actually, the Tana Delta has 2. So this particular candidate is what we call the Protopterus aethiopicus, a very handy fish, a fish that can survive out of water for a period of up to 4 years. Reason being that they have primitive lungs. Yeah, this is another candidate that we found in the lower region, Oreochromis leucostrictus. It is not endemic in this region, but very common actually in the Kenyan rivers at present. If you go to Lake Naivasha today, actually this has outdone even the Nile tilapia. It's establishing very well in most of the Kenyan river systems. This one, Cynodontis, in the family of the squeakers. You'll find this in the Lake Victoria region and also in the Tana River system, especially in the lower part. This one, Lacustricola bucobanus, they are called the lampayes. And recently, actually, there's a new species found in the coastal region around Mkurumunzi, where a lot of mining is going on. This one is a very good aquarium candidate and is known to very quickly establish itself even in areas that you think that they are no longer found, mainly because they leave their eggs even in very dry seasons, and the eggs hatch immediately the rains come back. Yeah, so this one, Shilbe, very well known because of the continuous anospine, this one here, but also very common in many parts of the country. Now, important to note is that the fish species diversity actually and biomass increased from high in the forest as you move towards the river mouth. The diversity around the mouth forests and the areas slightly below was very low, basically because the water temperatures were low and many fishes actually will avoid such kind of environments. In other parts of the country, especially in Mount Kenya, the Aberdares and Mount Elgon, the rainbow trouts were introduced in the early 1900s. How they escaped the mouth forest, we do not know, because we do not find the rainbow trout in this part of the country. Now, all that data that I showed you and all those pieces were updated on GBIF through this platform, and this is the link. And I'm very happy because the lady that presented on the Saturday earlier on before I came here did this job. So she's done a lot of work actually trying to digitize data from the shelves to GBIF. So this was sponsored by JRS Biodiversity Conservation. Now, there are many, many threats facing the Mara River ecosystem in general. and by extension, the fishes of the Mara River. Earlier on, I don't know if I mentioned that most of the tourists visiting the Mara and Serengeti are less concerned about what is in the river, despite the river being the lifeblood of the Big Five and all the wildlife that you see in the two national reserves. So very few people are interested, and so very little action is done to save the water. On the upper reaches of the Mau forest, we noticed a lot of forest destruction, actually exposure of a lot of land. Most people are clearing this to get firewood and also clearing for areas for agriculture. So at the end of it, all this topsoil ends up in the river, and you know what that does to the river. Washing of motorcycles and cars, and even people bathing at the banks of the river, releasing a lot of pollutants and poisons to the river system. All this ends up in the fish digestive system. A lot of cattle, this photo down here, this is a watering point for cattle, and they will spend a quite substantial period of time here and release a lot of waste into the water. Aquatic biologists here will tell you that the waste actually use a lot of oxygen when it comes to decomposition, biological oxygen demand. That, in the long run, actually also affects fishes of the river. Now, in the papers, you've read a lot on fish die-offs in the Mara River. And majorly, this is attributed to what we call hypoxic floods. This mainly happens in the Tarek River, and that is why most of these deaths occur near the New Mara Bridge, just before the river exits into Serengeti ecosystem. So what happens is that due to a lot of grazing in the Tarek River, actually the upper part of the Tarek River, a lot of sediments are deposited into the river that are flushed down into the main Mara River. And it happens within a very short time. So once this water comes into the river, it flushes actually decomposing materials from below the water column up high above. In the water column actually includes hippopus, and you know hippos defecate in water. So all this waste is flushed upwards and mixes with sediments in the water column, actually giving what is called an oxygen deficit. So this oxygen deficit causes all this. A certain change of water with oxygen with water without oxygen. Yeah, so these are the same sediments I was talking about from the Tarek River. From this bridge here called the Emarti Bridge, you can see what comes in the river per second. And this is the index. And the tonnage per day. This is what comes into the river per day when there are flash floods. This is far much above before Tarek River comes in. So I want you to see how much the Tarek River brings into the river, as opposed to what you find higher up the river. So this is what the Tarek River brings in: 710 tons of sediment per day. So I think I spoke a lot about deforestation in the Mau forest. And this is what is happening. A lot of forest clearance for agricultural activities. A Actually, in the recent past, we've earned like a 30% loss of forest cover and a 200% increase in agriculture. This includes both small-scale and large-scale farming. A lot of increase in tourism facilities, and very sad to note is that Serengeti is 10 times bigger than Masai Mara. Serengeti has hotels and lodges next to the river, slightly above 100. But despite Kenya having an area 10 times less than Serengeti, and I won't shout, has close to 400 lodges and hotels. You can see the threat on the Kenyan side. I hope I want to be put into task because of that statement. Yeah. So, lastly but not the least on the threats, unsustainable fishing practices. I know most of you are asking if there are fishing activities in the Serengeti and Masai Mara. No. But in the Mara wetland in Tanzania, we noted that the fishermen, actually there were so many fishermen, that is number one. Number two, the kind of fishing that they were doing was use of ring nets that are illegal, mosquito nets, meaning that they used to harvest even the young ones that are supposed to be fathers and mothers tomorrow. Yeah, as I conclude, this is the team behind the research. These were the fishermen. And apart from fishes, actually, were other standards. So the team was big. We have entomologists in this team also. And apart from fishing, we were also doing water quality. We were also studying migration of the Lake Victoria label, and happy to notice that Masaira being an attraction for migration of wildebeests, the other migrants, but not in land this time, but in water. We are still doing this, and I'm happy to report that we realized that we noted that the fish travels between 75 and 90 kilometers upwards of the Mara River. Actually, this is longer than even the wildebeests migrate. So, Asante, this was our other part of the team during the research. Thank you very much.
Thank you, Edward. And that brings to the end of our presentations. Now we get into a brief session on questions and answers. And I wish, can I request the presenters who are maybe behind to just come closer, and then we get, because I can see Kireka is there, Beatrice is there, Lovisa is there. Who was Jag is there, was obvious. Yes. Can you come close? And then can I get questions? I hope you had noted them for, because the promise was that we do all the presentations and then have a session for questions. And then you also make sure that you direct your question to the presenter so that we can have some, they can address them accordingly. We'll start from there.
Greetings to you all.
Greetings.
My name is Emmanuel Busera. I am a researcher at the Afro-Asia Institute of Strategic Studies, and I'm also the co-editor of the Patriot Speaks journal, and we practice some environmental journalism, writing about some environmental matters. Our very brilliant panelists, Paul, Zhang, Jacob, Veronica, Judith, Lavisa, Beatrice, and Edward, have actually given us master classes on biodiversity and conservation. But my first question goes to Zhang. I don't know if Zhang is in the room, or I don't know if he said he's Pei or Zhang, whatever, the one from the Chinese Research Institute.
Yes, exactly.
I know China is a very big country, population-wise. And now to save biodiversity, you know, people rely on plant species for energy for cooking. They use them as utensils. Some species are used for medicine, etc., etc. Now, how are you blending or fusing your science and the traditional systems to ensure that as people rely on these species for their livelihoods, they are also observing the science that you are bringing to them in terms of conservation? My second question goes to Jacob. I think Jacob, the one who gave the talk on the tortoise, very interesting, wonderful presentation. Now, you talked about, in your analysis, the test for circular uniformity, you said you are using the Rayleigh test. What really informed your choice for the Rayleigh test? And my last question now goes to anybody on the panel. You know, science, we know is a systemic enterprise that organizes and builds knowledge. And every time you test the knowledge, you produce the same result. So then science is the art of proving that knowledge is right. But when you look at the mind of a scientist, they say anything that you cannot observe and measure does not exist. But scientists brought us the formula of infinity, and we learned it in elementary calculus. You add something to it, infinity. You remove, infinity. It has no beginning, no end, but still scientists brought the formula. Now my question is, when you are coming to do your research, because I've seen you have done wonderful presentations, methodologies, test keys, and whatnot, you are meeting our traditional scientific systems that have existed since before Christopher Columbus lost his map. So when you come to the global south, you are not headed into an empty space. You are actually headed into an already crowded room that has got a system that has operated successfully for years. Now, for people to apply your science, what methodologies are you employing to ensure that the traditional people with their scientific methodologies that have been there for so many years don't feel alienated when you come with some alien words that you are calling simple species, and they have their own kind of way of doing things at the local level? In a simple way, can you explain to me how you will involve the community so that there can be a general uptake of science rather than the paternalistic way that the West looks at our traditional scientific systems? Thank you.
And I'll suggest you just be brief and direct to the question because of interest of time. We have got a poster session ahead, and
Um, My name is Chintan. I'm a PhD candidate at LMU Munich. My question is to Dr. Najag. You talked about the Mara River fish biodiversity. I'm also working around that area, and I have a question that, are there any bioindicator species as you go down towards the mines and towards the Mara wetland?
Thank you so much. So my name is Odera George. I am not a researcher, so I'm a practitioner, a representative of the village people. So my first question is to, you talked about what you call taxonomic uncertainty, right? Just recently, I felt very stupid somewhere. I don't want to say where, because my friends are in the room. So at some point when I was a student, there were acacias. In fact, you actually took that from me. So I knew we are talking about acacia species, and we were discussing. Now, recently I went there and it turned out to be Vechelia. How do we deal with taxonomic uncertainties that make practitioners look very stupid? Especially when you realize that you learned this and it is now this. In the course of time, you're not even aware what happened. So what is your suggestion in terms of addressing taxonomic uncertainties? Because to be very honest, I dealt with it in a clever way. I did not pretend to have known, so I escaped the embarrassment. Dr. Edward, I have seen a presentation that you have indicated very significant threats, particularly to the fishes, looking at very obvious things like washing vehicles and motorbikes. I'm certain, and this is part of the reason that has brought me here. You're not just seeing this because you are interested in seeing problems. So how do you link addressing some of these issues, especially that National Museum is a government entity, looking at the overall role that the Water Resources Authority has in terms of addressing this? So how has the linkage been between your studies, but most importantly, within the policy area, or where the Water Resources Authority should come in to address such kind of challenges? And lastly, again to you, you said something, and somebody sitting behind you should have asked this question. There's somebody right behind you. So you showed a picture of the cattle of drinking water in the river, and you said many of them defecate, and it takes a lot of oxygen to decompose that, therefore threatening the fish. Now, you are a researcher. Before you can be accused by the community as being insensitive, how do you then balance the need to protect the oxygen and the fish and everything? By the way, I'm a Luo, so I love fish. But how do you protect that? How do you balance that interest with the overall interest of the community, the pastoral communities, in keeping their livestock? Because where else, what is your suggestion? Where else do they graze their, do they water their livestock? Thanks.
Yeah, go ahead. Yeah, I think so.
Hello. So first of all, I think I will help with addressing your question on acacia and the taxonomic uncertainties. I also have a question towards Zhang, but what happened with the whole acacia story, Senegalia? First of all, my name is Joseph Mburu. I'm a master's student at the University of Oslo. I'm working on phylogeny of aloes. So what happened with the acacia story? There are normally rules that go into naming, and there are always taxonomic revisions. So there was a conference happening in Australia, and the conference did not have a lot of taxonomists from Africa. And the oldest name, which is normally the one which is adopted, was the acacia was the one which belonged in Australia. So that was what was adopted there. But we did not have more taxonomists to be able to fight for acacia to retain in Africa, because a lot of people know acacia from Africa. And that is what you have in mind when you think about the Mara and all these other places with acacia and the Commiphora. So we do not have people there, but you can still use acacia in day-to-day use within Africa, and that's what is also being adopted by like my supervisor when they are in Africa. So that it's what happens with the old story with Acacia. So we need more taxonomies to be able to fight for us. And there are other there are places where you can have some kind of like balance. Previously for like a family like Asphallidaceae. The oldest name was like in Australia, but we had a lot of people who fought for that because that whole family for aloes was in Asphallidaceae, and that was retained because to not have that kind of confusion. So we need more people, and all the researchers, I would say taxonomy is a very interesting place to look at. So I think that should address that, and maybe should also add something on the same. My question is towards Zhang on the phylogeny that he has talked about. And what analysis did you guys run for your DNA phylogenies, maximum parsimony or Bayesian analysis? Also, I'm interested in the kind of outgroups that you use in Balsaminaceae. Which outgroups did you use, and did you include any barium specimen for the DNA samples? Like, did you have any DNA sample specimens from your CP phylogeny and the DA phylogenies? Yes, I think that's it. Thank you.
Last one, and then we go ahead.
Okay, thank you very much. My name is Jeff Uchieng. I'm a student at Marara University. I'll be brief. I study ecology. So my question goes directly to Dr. Paul Kirika. I hope he's around because his research was so interesting. First of all, because he sampled from my home area in Migori County, and also because one of his findings, the orchids, is related with the Sphingidae, the moths which I'm studying. So my question is, you sampled up to Thimlich Ohinga and also Mukuro. You noticed that they're about 50 kilometers from the shores of the lake. And since I come from the area, you realize that around the lakeside there are several hills that are very hard to reach, which means they have not been affected by anthropogenic activities, which means they might be holding certain species that probably you are not able to reach. So I want to ask you to how are you going to address that with your research? And then you are able to discover some orchid. I'm interested to know which particular area you discovered this orchid, because orchids are endangered, but there are also some that are pollinated by certain hawk moths, like for example the Agrias convolvuli. So I would want to ask if you would be kind enough to maybe tell me where this orchid is, so that I will be able to, if I'm interested in doing further research on my Sphingidae, to see if they are available there. My research on hawk moths around Ruma National Park, that is not far from that area, I discovered 8 species of hawk moths, but I did not find this particular one that pollinate the orchid. So if you would be kind, I would be really appreciate if you tell me where you find this orchid. Another question goes to Ms. Veronica Onduso, who was doing spatial temporal patterns of small mammals, mammalian communities revealed by owl diet in Amboseli. One of your findings was that, according to your finding, you realized that there was an increase in species diversity over time in both of your study areas, but you also observed a decrease in dominance. So I wanted to understand this dominance. Did you find it in the larger mammalian species or the smaller ones? And what is this implication on your study? Thank you.
Okay, thank you. Let's start with Zhang Jing.
Thank you for your question. Firstly, I will answer the first question. When we conducted field works in the wild, we will frequently contact with the local people and learn from them about the ethnobotany knowledge. That can enhance the understanding of these plants we are working on, both us and the local people. And we both can also realize the importance of those plants and better to conserve and understand and utilize those plants. And for the second question, we use ML and BI methods in the phylogenetic analysis. And for the materials using in this study, we use Qiaojin DNA kit to extract the DNA from the dry specimens. It can even extract DNA from specimens long before. And we use the normal DNA kit to extract the DNA from our silica gel dried samples. We also use multiple sections on GenBank to establish the structure, the basic structure of the impediance trees. Thank you.
I hope the questions have been addressed. Let me pass it to Jacob.
Thank you for the compliments and the tough question. He asked about what informed me in the use of relay tests, to address the issue of homing. And I would say that first it was a challenge because I was in the field and this one came as a coincidence. It wasn't something I had planned. And when you want to publish, you sometimes meet tough reviewers. And so I met one of them who declined my paper, but another one was kind and recommended something. And so relay test is all about circular statistics. Remember, this homing animal, you are not continuously following the animal moving all over. You measure, or you go and take the coordinate in the morning, perhaps in the evening or the following day. So you are dealing with coordinates, and you want to make something out of that. So you want to come up with a vector joining between one coordinate and another. And that is why what necessitates the use of circular statistics. And so I don't know many methods in circular statistics other than this relay test. And we have another one called V-test, which is just a modification of the relay test. And the issue is you want to see, these animals are moving. Remember, some are going to the east, to the north, others to the west. You want to see if there is a consistent pattern. If I was to answer in summary, that would be my answer. You want a test that will be able to create meaning out of all this haphazard movement by the animals. So,
that is it?
Yeah, I want to say that
I hope
sorry, maybe the issue is now there was a clear pattern of movement, but you could only, as from the results I presented in the morning, we had 13 animals moving in different directions. But we wanted to know, is there a clear pattern of movement? And out of this relay test, which takes into consideration the number of movements, like from this coordinate to the other, you want to take an average of that and the mean, resultant, vector from each one of them, then you realize not all of them had statistically significant homing direction or orientation. But then 2 of them have some clear pattern, and you would perhaps give up. But when you combine all of them and then calculate a pooled Rayleigh test statistic, then you realize there is a pattern, even though it is not very strong. But I had answered your question. Thank you.
Okay, let me, because of issue of gender, let us have a lady first.
Okay, so the 2 gentlemen, thank you for the question, but that's a very tough one. Myself, I keep asking the same question, so I'll try to answer it. I'm not a taxonomist, but I'll try to answer it, yes. So, especially with the acacias, this is an ongoing debate. I don't think it's something that will end or that will come, that the taxonomists will agree on one thing ever. So if you are a non-scientist, or I would advise that you leave out the taxonomic debate. And honestly, since you have been learning about acacia, myself, I'm still stuck with using acacia. I haven't accepted the Vachelia and the Senegalia until now. So it's up to you which name you want to use. And it would be nice if you are working with the community to educate them on the common names instead of using scientific names. Okay, so, but no matter how many times they may try to change taxonomy, conservation actions should still just continue. Okay, so so now you asked what happens or what's the way forward with the issue of acacia being renamed to Vachelia and Senegalia. So this is something that the debate started after they did the DNA evidence, whereby the acacia in Africa have thorns and the acacias in Australia don't have thorns. So after they did the DNA, they found several distinct evolutionary groups, like evolutionary DNA results. And that's how the renaming of our acacia to Vachelia and Senegalia came in. So it's not that our scientists didn't fight. I have an expert that comes to our institute here and then, and she keeps saying, these international experts are giving me migraines because they are forcing us to use to use what they have decided for us, although the species are in our countries. So, but the decisions are not really up to us. We can fight as much as we want, but if we don't have enough support, there is nothing that we can do. So after the debate, they had to do a voting process. So the results that come out with the highest voting and the highest international support wins. So that's how we lost the debate. I hope that helped.
Yeah. Yeah, I hope I'll be able to wade in into that issue. Thank you for your 2 questions. One is about some areas in the Lake Basin, Lake Victoria Basin, that are, as you say, intact, and that we did not sample. And this is for the reason that resources are always limited when you go out and you have to be focused on the area that you want to study. And for this case, we were studying 3 counties, Migori, Kisumu, and Homa Bay, and we didn't visit Siaya. We had intended to carry out some work in Gwasi Hills, which I guess are the ones you wanted to ask. But the local communities were very happy with us. We were a large number of, we were a big team of various experts, including insects and mammals and other organisms. And they did not allow us to sample the hills for some reasons. And when we tried to find out, it's like there is also a lot of encroachment in the hills, and also cultivation of some illegal plants, mainly cannabis. That is what we came to realize. So the locals did not want us to sample that area so that we don't bring this out to the public, because they thought we were government officers. So we were not allowed. We would have loved to sample Gwasi Hills, but we were not able, and we hope in future we will be able to sample. The second question is on the orchids, where we found the orchids. We actually found only one population, and this was in a farmland in some remnant indigenous trees. This is an epiphytic orchid that grows on other plants, on trees. And it was, we only found one population in a farmland. I'll get you the coordinates if you are interested to follow it up. And we managed to collect a small sample for the Botanic Garden, for conservation at the Botanic Garden. There is a lot of removal of those indigenous trees in the area, and this has affected the diversity of the orchids, which are the epiphytic orchids, and even other organisms. I hope I've answered your question. If you are interested, you can we can have a chat, and I can give you the location. The other thing, do I want to wade into the controversy taxonomic changes that happen? This is, it's annoying, but it's unavoidable, because as new knowledge come into as new knowledge is generated, there is need to make some changes. For example, over the last 10 or so years, or 20, there has been a lot of molecular work going on, and this has brought new knowledge, and molecular sequences have been, and molecular data is now used largely in taxonomic work as a character. And this has brought a lot of changes that are happening, and it's annoying, but you cannot avoid it. So but you can continue using the old name because what in taxonomy is called a synonym. So you can still be able to find out what that species is. So don't labor so much, but yeah, because these changes will come and they are unavoidable. Thank you.
Thank you very much. I think some few questions were directed to me, and I'll attempt to contribute a bit on them. I won't talk much about taxonomy, mainly because the debate is the same. I remember last month I was making a presentation somewhere about some species that we caught somewhere in the Kilimanjaro Churu. And at the end of the presentation, one of the species that I was presenting about had a new name. So these will keep on changing, especially in fish, where we have slightly over 40,000 species. And most of them keep on being lumped together. At times, other times they are separated. So this debate will never end. Someone asked about bioindicator species in the Mara River. Yes, they are there, especially those that were found in the upper reaches of the river and not found downstream. Basically, in any actually river system, especially those that emanate from forests, the waters leave the catchments very clean, but as they move downwards, actually they are laden with sediments, pollutants, and all forms of threats. So most of the fishes that were found on the upper reaches, we would say are the indicators. Some of them include the elephant snout fish. Actually, this is the candidate that I spoke about being mostly affected by a drop in the levels of dissolved oxygen. Yeah, there are others like one of the chiplinins, actually that we found high up one of the very pristine areas in the forest. We actually caught only 2 specimens, and the last records, actually before we caught them, was in the early 80s. So it means that they are not able to traverse the river downwards, basically because of their sensitivity to environmental change, especially temperatures. Now, my colleague spoke about threats and spoke about how we balance cattle defecating in the river or in the areas that they take water, and how we balance that between them and fish. The truth of the matter is, this threat is big. Because I know the other question that you are not going to ask, that you are going to ask if you had enough time, was if we are blaming the cattle, what do we do to the hippos that live in the water? Now, I will only say that every small action counts. There's nothing that we can do to the hippos because that is their natural habitat. They will never leave. But that small action by the cattle, by building designated areas for them, that will control the little impact that they are taking into the river. Though every small action counts. So that is one of the remedies that I mentioned. There are many others that we will discuss in another forum. Now, you also asked how, despite shouting too loud about all the threats facing the Mara River and the Mara fishes in general, what are we doing to connect with the communities and other stakeholders? Yes, it is true that we've been shouting very loud, and we are actually going to shout even louder, because if we don't, then we are going to lose a very important resource, that is the Mara River. We are going to lose revenue for Kenya and Tanzania. We will no longer see border-based migration, and probably the Masai Mara will cease to be, I don't know, the seventh or eighth wonder of the world. We do not want that. So, in the course of our research, we did not do it single-handedly. Actually, we involved a lot of stakeholders. This research not only involved fish, but also macroinvertebrates, and how all these land-water linkages relate and how they intertwine. Because all the actions that happen in the terrestrial environments have a direct impact to what is happening in the river. Number one, we've been working with what we call the Water Resource Users Association in the area, basically based in Narok and Bomet counties, around 10 of them. And we have been going around sensitizing them and even working with them. Actually, most of these sampling occasions for fish and macroinvertebrates were together with stakeholders. We did not leave them behind. As we speak now, one of the RUAs, called the Mara RUA, that have offices near Mulot town, have actually constructed a research center. We trained them how to monitor water health using macroinvertebrates. I don't know if my colleague Laban is here, but he has been leading that component. So they are using an index called SARS, the South African. I saw Professor Esther Kyoko here. She knows better about the SARS. That the absence or presence of certain macroinvertebrates will tell you if water is getting better or worse. We've trained them how to use that, and they are doing them themselves. So they are able to communicate between themselves and sensitize their fellow members to avoid some actions that make the water worse. Secondly, in the Standing area, both in Narok and Bomet, we worked with local primary and secondary schools. The aim was to sensitize them on the importance of river conservation and environmental conservation as a whole. Actually, we gave presentation in like 12 schools. Apart from the local primary schools, we gave actually talks to 5 universities, starting with Masai Mara and several other universities. We conducted 2 trainings, one in Nairobi here at the National Museums of Kenya, and another one at the University of Eldoret. That was to ensure that we also sensitize our future leaders in environmental conservation. Last but not the least, Kenya and Tanzania have a joint initiative that is aimed at conservation of the Mara River, from the catchment to the mouth. These are joint efforts through various stakeholders to ensure that all is not lost. Because as Kenya, if we destroy the Mau forest, our neighbors Tanzania we will have no Serengeti. So it affects all of us. So we have the Maradi that is celebrated every year on the 15th of September. And it's in Kenya this year, the following year it's in Tanzania. So we alternate. The Mara project was in Butiama, Musoma last year. And we were able to give out our results to all the stakeholders from both sides of the region. We gave our recommendations. All the leaders were there. Leaders from the East African community, the Ministry of Water, Minister for Environment from both countries. And we made sure that our shouts were heard. At the same time, in December 2025, driven by the mass destruction of the Mau forest, our shouts were heard by KenGen, the Kenya Energy Generating Company. And together with the Mara Project and the National Museums of Kenya, we contributed in rehabilitation of the Mau forest, actually by planting, together with the local Ruas, 25,000 trees. This was just one of the initiatives, and many more are coming. Last but not the least, and very important to you all in this hall, the National Museums of Kenya, in our galleries here, those that have visited the exhibitions, our exhibition is in the Aga Khan Hall on the first floor. We launched it on the on the 4th of March, and it will be running until March 3, 2027. So it will be there for 1 full year. The target audience is 2 million visitors, including school children, tourists, and anyone else that comes into the museum. By this, because we've really simplified it, including our resource and other, and the impact of forests, destruction, pollution, and everything that happens to rivers, not only the Mara River, but all rivers in general. The aim is to inculcate knowledge to everyone, including primary school going kids, so that we inculcate that knowledge of conservation from the young age. Even as they grow up, everyone out there will see that this river is important because of A, B, C, and D. So I'm requesting that you all visit the exhibition in the course of the week. Asante.
Thank you, Jackie. I think he has addressed more than what was asked. But anyway, he appears to be a politician. After.
instance in survival, the mastomys databases now coming in, which is disturbance rights, which means the community now, the social design, the guide, the next complete design around favoring the general species on time. That's what I don't know what I wanted to do.
Okay, thank you. Yes.
Okay. Okay. I can't remember his name. The gentleman in blue, he asked the question to all presenters. And if I'm getting his question right, he said the scientific systems that scientists use, those are methods that a person needs to have scientific knowledge. So you said that what systems can we suggest which can be applied by people at community level, especially indigenous people, who are not scientists and who don't have scientific knowledge. Is that the right question? Okay, but if we are being honest, scientific knowledge and local or indigenous knowledge complement each other, and that's when conservation is strongest. So you can do scientific research without including local knowledge. If I have to point out a few methods or systems that we can use to include community or to educate community, and they can get an understanding of scientific research, then that will be things like participatory approaches, whereby as a scientist, as we go out in the field to collect data, do research, we can invite community or indigenous people that are interested to take part in the research. And then our methodologies are not that complicated to collect data. So you can explain to an indigenous person, especially if you speak the same language, they will be able to understand it and they will be able to help you with data. So what we do back in Namibia, especially for the JRS project, when we go out in the field, that's in the southern part of Namibia, most of the land there is farmland. And for you to go in those farmlands, you need permission from the farmers. So we write to them, we explain to them what are we coming to do in their area, what findings are we going, what results are we getting out of doing that. And then as we go to that area, we try to bring all farmers together, and then we educate them on these things. Those are areas with our endemics. So we have come up with a way that we convince them to allow us to install the Earth Ranger app in their phones, and they accompany us in the field, and then we take records and we ask of them to keep monitoring for us those records, as we can't be there all the time. So we can track the records from the office while they are the ones doing the monitoring in real life. And then another approach is citizen science, iNaturalist. We put up, challenges, citizen science challenge, whereby we say any citizen of Namibia, wherever you are invited to take part in this challenge, and the citizen that has the highest records gets a prize. So that's one way of bringing people into science. Another thing is environmental education and awareness programs. For our country, now that we are experiencing high incidence of plant poaching, we have incorporated our traffic officers, border officers. We have printed out pamphlets to educate the officers by those border posts or by the roadblocks about the certain species that are vulnerable to poaching. Because the poachers, they go in the field, poach, they come at the border post with a fake permit because the officer doesn't know what plant that is, what is important. They say, okay, you have a permit, pass, go. So we have tried, that. If anyone wants to add on the question,
okay.
They are welcome.
So thank you, Officer. And thank you very much for the patience and support you have given to me and my team. The rapporteurs are there. They are still here. Sorry? Oh, okay. Okay, so we are going to have a break. There are two presenters who you did not, there was no question, but maybe some. And possibly, even tomorrow, you know that there's Beatrice, and then there's Judy. So specific questions, you can just see them afterwards, or even tomorrow. Then at this juncture, we'll go for tea break and then after that, at the same time, we have a poster session. They are just outside where we'll be taking the tea. So thank you very much, and have a nice evening. Oh, and sorry, I've just been reminded. Tomorrow we are meeting here at 8:30.
Yeah, we'll meet here at 8:30. So let us keep time, please. Okay, you are free now to go for the health break. And then don't forget to go through the posters. They are there for you, and I hope the owners of the posters will be there.
Hii tu ndio inafanya. Umezima?